Now we are going to learn the structure of eukaryotic cells. So the cells is it is very essential to study the cells because it is the simplest form of the life, and for each and every organism, cell is the basic structural functional unit of the life. The cell, this term is named by Robert Hooke in 1665 after observing the Koch cells under microscope. Now the cell theory. Schleiden, Schwann and Rudolf Virchow proposed the cell theory. Schleiden and Schwann proposed old cell theory, which is all living things are composed of cells and product of cells. Cells are structural functional unit of the living organism. Whereas Rudolf Virchow updated this old cell theory, which is new cells arise from or new cells comes from the pre-existing one, that is omnis cellula e cellula. Microscope is one of the advanced technology that we have to use for the observation of microscopic cells. The simple light microscope was invented by Anton von Leeuwenhoek in 16th century, and first time the structure that he observed, he gave the name animalcules. Now we are going to learn cellular organelles. Nucleus is one of the very important cellular organelle. It is a control center of the cell surrounded by the nuclear envelope. It contains DNA. Nucleolus, it is a site of ribosome synthesis. Cellular organelles also include ribosome, which is a site of protein synthesis. Free and bounded type of ribosomes are present. Freely floating ribosomes inside cytoplasm prepare or synthesize the proteins for the use inside the cell, whereas the bounded ribosomes to endoplasmic reticulum make the proteins which used and transported out of the cell. Then cellular organelle include endoplasmic reticulum also. Endoplasmic reticulum made up of membranous tubules and cisternae or sacs. Smooth endoplasmic reticulum and rough endoplasmic reticulum are the two types of endoplasmic reticulum. Smooth endoplasmic reticulum, it is because there is no ribosome attached to the membrane. Smooth endoplasmic reticulum helps in synthesizing and transport of lipid molecules. It also controls glucose and glycogen conversion inside liver and muscles. It helps in detoxification of drugs and other poisons. Sarcoplasmic reticulum, like muscular endoplasmic reticulum, stores the calcium needed in muscular contraction. Rough endoplasmic reticulum, to which ribosomes are attached, these ribosomes are of 70S type, and it helps in synthesizing and transport of proteins. These are the structure of smooth endoplasmic reticulum and rough endoplasmic reticulum. Now Golgi apparatus. So the product of endoplasmic reticulum are modified and stored with the help of Golgi apparatus. So Golgi apparatus helps in modification and packaging of the proteins. Lysosomes are used by the cells to digest the macromolecules. These are the sacs of hydrolytic enzyme which helps in programmed cell death. Programmed cell death is apoptosis. Usually it found in the only animal cell. Vacuoles are various type, including food vacuole, contractile vacuole, central vacuole. Food vacuoles store the food. Contractile vacuoles pumps the extra water out of cell in case of freshwater protist. Then central vacuole is present in the plant cells, which occupies 3/4 area of the plant. It stores the organic compounds and inorganic ions and water also. It is also known as a tonoplast. Peroxisomes contain the enzyme that transfers hydrogen from various substances to oxygen and produce hydrogen peroxide as a byproduct. The function of peroxisome is to break down the fatty acids into smaller molecule for the cellular respiration and detoxification of alcohol inside the liver. Mitochondria, it is a powerhouse of the cell. It is a site of cellular respiration. Adenosine triphosphate or ATP get released or get synthesized inside the mitochondria with the help of or by the breakdown of organic molecules. If more metabolic activities are present in the cell, that means that particular cell require more amount of mitochondria. Chloroplast is present only in plant cell. It is a site of photosynthesis. It contains the green pigment known as a chlorophyll. Along with that, chloroplast structure contains grana. This grana is a stack of thylakoids. Thylakoids are the coin-shaped structures which arranged one above other. Stroma is a fluid-like substance which is present outside the thylakoids. So thylakoids or grana, it is a site of light-dependent reaction, whereas stroma is a site of dark reaction or Calvin cycle. Cytoskeletal mainly gives the shape to the cell. It is responsible for the movement of cell and movement of organelle within the cell. It is made up of three types of protein fibers, that is microtubules, microfilaments and intermediate filaments. Centrioles only found in animal cells. These are visible only during the cell division. Centriole contain very simple 9+0 arrangement of microtubules. It may give rise to the cilia and flagella also in few organisms. It also involved in the formation of spindle fibers in the animal cell, but it doesn't play that role in case of plant cells. Cell wall, it found only in plant cells which protect and maintain the cell shape. Cell wall also prevent the excessive uptake of water and hold plant against gravity. Cell wall in the plant, it is made up with the cellulose. Primary cell wall, it is very thin and made up with cellulose, whereas secondary cell wall is thick and found in the woody plants only. So in this way we have studied the eukaryotic cell. We have also studied the difference between prokaryotic and eukaryotic cells. So the major difference that present in the prokaryotic and eukaryotic cell, it is a present of eukaryotic cells consists of centrioles. Eukaryotic cells also consist of mitochondria, chloroplast. Microtubules are also present in the eukaryotic cells. Nucleus, well developed nucleus is a main speciality associated with the eukaryotic cells, whereas prokaryotic cells does not contain these kind of well developed nucleus. Nuclear membrane is completely absent in case of prokaryotic cells. Prokaryotic cells contain cytoplasm in which double-stranded circular coiled DNA is freely floating inside the cytoplasm. In case of prokaryotic cells, flagella is present as a external appendages. Also, external appendages contain flagella, pilus and femori. Cell envelope, in case of prokaryotic cell, it is made up with the capsule, which is outermost layer. Middle layer is the cell wall and innermost layer is the plasma membrane. Inside to that cell envelope, cytoplasm is present. In case of prokaryotic cell, 70S type of ribosomes are present. Whereas in case of eukaryotic cell, both 70S and 80S type of ribosomes are present. These 70S types of ribosome in case of eukaryotic cells are only present inside mitochondria and chloroplast. Mitochondria and chloroplast inside the eukaryotic cells are self-replicating units. Self-replicating units means during the cell division, at the time of cell division, these cell organelles are not dependent upon any signal given by genetic material present inside the eukaryotic cell, because they have their own DNA, own circular coiled DNA is present in the mitochondria and chloroplast in both the structures, and this DNA generate the enzyme which helps for self-replicating. Ribosome of 70S is present in both mitochondria and chloroplast. Because of that, by using RNA, these two structures, these two cell organelles can synthesize their own proteins at the time of cell division, at the time of self-replication. So these two structures are absolutely not dependent upon cell signal. So it is all about the prokaryotic cells and eukaryotic cells.